All right. First of all, I'd like to thank the organizers for giving me a chance to present our work. And I'd also like to point out the change in the title. It was correctly pointed out to me that gene regulation means a very specific thing. So I'm going to talk about modeling gene expression dynamics during cell cycle. Uh, how can I use the laser pointer? Okay.
Use this one.
Oh, no.
You can also switch the slides.
Yeah, this one. Okay.
Hello, Romana.
Perfect. All right. So cell cycle and cell differentiation are two of the most fundamental biological processes. Here I'm depicting them with a very simple schematic diagram. So cell goes through different cell cycle phases. In G1, it just grows in size. In S phase, it synthesizes the DNA. In G2 phase, it corrects for some of the copying errors and prepares the cell for mitosis. And in mitosis, the actual cell division happens. And sometimes the cells enter a quiescent phase, so it's G0 phase. During this phase, the cells are not cycling. And here I'm again depicting a very simple schematic diagram of a stem cell differentiating into different types of cells. I'm not showing all the lineages, obviously. And what I want to point out is there's a complex interplay between these two processes. So what we do know is the stem cells, they cycle really fast, of the order of hours, which is much faster than normal mammalian cells. And during their differentiation trajectory, they slowly lengthen their cell cycle. And terminally differentiated cells are not cycling at all, so they enter a quiescent phase. And on the other hand, in order to activate a differentiation program, you need to be cycling, and specifically in G1 phase. So there's this interplay, a dynamical interplay between these two processes, and this is one of the main research axes in our lab. Today, I'm just going to focus on the cell cycle bit, okay? And particularly, I'm going to talk about the gene expression dynamics during cell cycle. So to do that, I'm going to introduce RNA velocity as a framework to study cellular dynamics. So we all know the steps involved in transcription. You first make unspliced RNA, you remove the introns, you get spliced RNA. This is what codes for the proteins. And then once the gene is no longer needed, it gets degraded. This is the same dynamics in a graphical form, so I'm not going to go through this. You can also plot this spliced and unspliced on a phase diagram. And you can see that the gene would follow this trajectory. This is the on phase of the gene, and this is the off phase of the gene. And this is the steady state. What I would like to point out that in typical gene expression data, you get a snapshot of the system. So let's say you have a gene with the expression level about that much. Just by looking at this, you can't say whether the cell was here or here. But now if I give you the unspliced levels also, then you can tell me whether the gene is on or off. Okay. So that's the basic idea behind RNA velocity to infer the dynamics. This is the system of coupled ODE to represent this dynamics. I'm not going to go through it. I think everyone here is familiar with ordinary differential equations. Here's an example to how to demonstrate the use of RNA velocity. So here we have bulk RNA-seq samples of mouse liver cells at different time points during their circadian cycle. And we're looking at the unspliced and spliced levels of circadian genes. Okay. So now if we do a dimensionality reduction and plot this on a PCA map, we can see that they nicely follow on this circular trajectory. We can also solve this system of ODEs for all these genes and get alpha, gamma, and beta. And using the current state, you can now, because this is a dynamical system, you can predict the future state. So that's what is demonstrated That's what's depicted by these arrows here. Furthermore, now if I give you a new sample and I don't tell you what phase it belongs to, I don't give you this hour information. Using this PCA map, you can tell me where it is in its circadian phase. Okay. So we wanted to ask, can we do the same thing for cell cycle? Instead of circadian genes, if I give you the cycling genes, and instead of the circadian phase, I give you the cell cycle phase of all the cells, can we make this map of expression to the cycling phase? So to do that, we did the following experiment. We cultured mouse embryonic stem cells in this LIF2I medium. This basically prevents the cells from differentiating. So all they're doing at this point is cycling. And it takes about 14 hours for them to go through one cell cycle. We did single cell RNA-seq profile of these genes, of these cells, excuse me. And then this is what we get. So here I'm showing you the unspliced and spliced levels of a gene called NuSAP. There's a bit of data processing that goes before arriving to this step. NuSAP is a gene that codes for a protein that makes the mitotic spindle. So it's very important to have this just before you go into mitosis. So by looking at this phase diagram, you could say that these these cells, by the way, every dot here is a cell. So these cells here are probably achieving are in the G to M transition. We also know that you don't need NuSAP when you're in G1 phase because you're not going to do cytokinesis. So these cells here are probably in G1 phase. And you can get a sense of cycling trajectory by looking at this spliced and unspliced levels. Now, the thing is, we don't just have NuSAP, we have a bunch of other genes. And so we have to get a consensus of all of these genes, use all of that information in order to predict the cell cycle phase. So to do that, we developed a tool in our lab called DeepCycle. What it does is it looks at the unspliced and spliced levels for all the cycling genes. It's an autoencoder, so we reduce this to one latent dimension, which is the transcriptional phase, the theta. This is the pseudo time of the cell. And from this one latent dimension, we're able to reconstruct the entire expression. Here, just demonstrating how this is used. So for every cell, you predict a transcriptional phase theta, and then you sort the cells by that phase. And you can see this oscillatory dynamics for CCNE. CCNE is a gene that is required for G1S transition, and you can see that its levels go high during that time. Similarly, you have the dynamics for CCNB. Okay, so we have this now, the expression dynamics, but what about the rates? What about the synthesis, splicing, and degradation rates? And particularly, can we infer these in actual units of molecules per unit of time? So to do that, we need to calibrate the data because the raw data that we get is not in terms of molecules, right? So to estimate the absolute counts, what we did was we look at the data. This is once again, every dot here is a cell. And then we calculate the moving average. So that's using a kernel smoother. So this is the solid line is the moving average. And remember that our cells are only cycling. They're not doing anything else. So if we remove the moving average, we're essentially removing most of the biological variability in the data. So what we're left with is measurement noise. This is a very strong assumption, but this is what we have. And so this now, because this is a counting experiment, the measurement noise should follow Poissonian statistics, right? So the mean should be equal to the variance. And so when we plot the mean and the variance, we see that they do have a linear relationship, but the slope is not equal to 1. So we use this slope information to recalibrate the U and S counts. And we think that that's the actual number of molecules in the cells. And we also did this for all the genes. Here I'm just showing the trend holds good. I'm not plotting all the data points. It's a subsample because there's just too many data points to plot. And so now, if we look at the total number of molecules, this is what it looks like. I was trying to dig up literature on this, and I could not find any that has a estimate for this number, which tells me that no one really does this type of estimate of actual number of molecules. So I'm curious to know what you think about this. I could find one reference on bio numbers, and they say that it's between 100,000 to a million, and our estimate is between those. It agrees with this. And on the right, I'm showing the distribution of U and S. So I take a gene and then I calculate the average across all the cells, and I'm showing the distribution of that. So here you see that the spliced counts are about 100 times more abundant than the unspliced. Okay, so this is the mathematical model. Once again, we have a system of coupled differential equations. Note that because this is a cycling dynamics, U, S, alpha, gamma, these are all periodic functions of theta. So we used Fourier series expansion to solve the ODEs. And we were able to get closed form solutions for all of these. Okay. So here I'm showing some exemplar solutions, and I'm demonstrating the diversity in the dynamics. So this we already saw, CCNE, this is the unspliced and spliced levels. We see that it has a peak during the G1S transition, which is what we expect. Now looking at the rates, we see that the synthesis goes up during G1 phase and then goes down during synthesis. And we see that the degradation more or less stays flat. Yeah. So the dynamics is mostly governed by the production of this gene. Okay. Whereas here, for NuSAP, you need to modulate both the production and the degradation to get this dynamics. And in case of V1, the synthesis more or less stays flat, and you modulate the degradation to get the dynamics. We did this for all the genes. Here's the explained variance. We put a threshold at 0.2, and we are left with about 1,800 genes out of 12,000. So this means that for most of the genes, the model is not doing well. These genes are probably not cycling, or we don't have enough data for this gene. It's because the measurements are noisy. Here I'm showing heat maps of the expression, synthesis, and degradation rates. And what we see is that we get waves of transcription during the cell cycle. Some genes are active during the G1 phase. Some of them are active during mitosis. The others are active for a longer period of time during the cell cycle. Here I'm summarizing the heat maps here. So what I'm doing is it's a distribution of theta at which you get maximum synthesis or maximum degradation. And once again, we see there's a very prominent production and degradation wave in G and S phases respectively. There's also another wave during mitosis. These are the global rates. So alpha 0 and gamma 0 are the zeroth coefficient of the Fourier series expansion. Basically, they are, when you just take the average of this trajectory, you get alpha 0. And so this is like the baseline production or baseline degradation of the gene. And you can see that at baseline, it makes about 0.5 molecules per hour. And the baseline degradation half-life is about 15 hours. The interpretation of this as splicing is a bit limited because this is confounded by intronic degradation. So this is not exactly splicing. Here I'm plotting the amplitude, which is alpha max minus min divided by 2 with respect to alpha 0, so the baseline. So how much do we get above baseline in terms of amplitude? And then this is ongoing work. We collected some data on multiomics. So this is single nuclear RNA-seq and single nuclear attack-seq for the same cells. And what I'm showing here is we get a peak in accessibility first, followed by the unspliced, and followed by the spliced, which is what we expect. Here I'm showing the heat map of all the genes in terms of accessibility. So there's a sharp decrease in accessibility during mitosis, which is what we expect because everything closes. And this is the average accessibility. Basically, this is the average of this heat map. With that, I'd like to acknowledge my lab members and the funding agencies and you guys for your attention, and I'd open it for questions.
Thank you, really nice talk. I was wondering, you showed with your deep cycle that you could, well, you were trying to find a consensus between the cycling genes, but do they need to be defined a priori, or do you have ways to look if in the structure of your data you can identify new genes?
Good question. So it's about how do you process the genes to find out which ones to take. So there's a pre-processing step that goes in this. You can find the details on GitHub. There would be a link to GitHub on this paper. But basically, we look at genes which have this bimodality in the distribution of U and S. Because if you don't have the cycling dynamics, then it's really not going to be useful.
So if you make the parameters in your ODE model periodic, are you not putting into the equations what you want to get out ultimately?
Yeah, so the periodicity is baked into the model. That is true. And we want to look at the cycling dynamics over the cell cycle.
I mean, you could use the limit cycle model and modify it, I guess.
Okay, what would that be? So
it would cycle by itself in a stable fashion, and then you could modulate it by inputs or whatever.
okay, good suggestion. I had not thought about that.
Because what you have is a linear system that